So today I'm going to give you an overview of cellular respiration and all the aspects that go into making ATP through the oxidation of glucose. So we're going to be talking about the whole aspect of cellular respiration. And as you know, there are 4 stages to cellular respiration. And the first stage is glycolysis. And so ultimately glycolysis takes glucose and through 10 steps it will turn it into 2-pyruvate. And during this oxidation, we have 2 reduction reactions that allow the body to store energy from this. So we're able to make 2 ADP and convert them to 2 ATP, and 2 NAD and convert them to 2 NADH. And these are net. And so after that, we then have the conversion of pyruvate to acetyl-CoA. And that's 2 pyruvates, 1 pyruvate per acetyl-CoA. And this takes 2 NAD and turns them into 2 NADH. So again, it is NADH here that is harboring our energy that we were making by oxidizing the pyruvate. And then finally, that acetyl-CoA is sent through the citric acid cycle. And in this process, we will end up generating another 6 NADH from NAD+. We will end up creating 2 FADH from FAD. And we also take 2 ADP and are able to generate 2 more ATP. So essentially at this point, glucose is fully oxidized. And along the way there, by that oxidation, we have to account for all 6 of those. So glucose is a 6-carbon molecule. Each pyruvate is 3 carbons, and since each acetyl-CoA is only 2 carbons, this means that each pyruvate lost a carbon dioxide. And since there's 2 happening here, 2 pyruvate to 2 acetyl-CoA, we've lost 2 carbon dioxides there. And then each of our acetyl-CoAs lose its carbon, it becomes oxidized, and so that carbon dioxide is going to be lost here, and all 4 of the rest of those carbon dioxides are lost. So by the end of this, we have fully oxidized our glucose. Now, if we are in the mitochondria, if you remember, the mitochondria has 2 membrane systems. We have the outer membrane, we have the inner membrane, and then we have the matrix of the mitochondria, which is within both membranes. Glycolysis happens in the cytosol, but the rest of this is all happening in the mitochondria. Now, the last step, oxidative phosphorylation, the whole purpose of that is to take these NADH molecules that we have generated and the FADH molecules and use them to basically make ATP. And so ultimately what we are trying to do is we are trying to create a proton gradient where we have a lot of protons within this matrix. We have high within the 2 membranes and we have low in the mitochondrial matrix. So we are trying to use the energy of these NADH molecules to make this gradient. And this happens through a series of complex molecules. We have complex 1, complex 3, and complex 4. So NADH comes and drops off its high energy electrons at complex 1, therefore becoming NAD+, and then that NAD+ is now available to go back and facilitate ongoing reactions of glycolysis. When those high energy electrons are dropped off, they are used, and complex 1 will pump 4 protons against the gradient. Then those electrons are transferred over to complex 3, which pumps another 4 hydrogens against the gradient. And once the electrons are done being used by complex 3, they'll be transferred to complex 4, and 2 more hydrogens are pumped. Thus, we have 10 hydrogens pumped per NADH. Now, the reason those hydrogens are important is because ATP synthase will use the proton gradient as protons flow down through. Every 3 hydrogens going from high to low is enough to take an ADP and convert it to ATP. So in this manner, all of our NADH and our FADH2 are able to create this proton gradient, and the proton gradient is in turn used to make ATP. That's how our NADH is used to make ATP. All right, so where does oxygen come in? At the end of complex 4, those electrons have to have a place to go. So what happens is at the end of complex 4, all of those electrons are added onto oxygen. All right? So if you have hydrogens, electrons, plus oxygen, you end up with water. And now we have removed the high energy electrons from complex 4. And by doing that, this whole process can continue to go on. More NADH can drop off electrons, they can go through these complexes, hydrogens are pumped, and at the end of it, the electrons are dropped off onto oxygen. Oxygen is the final low energy electron acceptor.